As soon as your photosynthesis and cellular respiration kit arrives, open it up and put the reagent refill pack in the fridge at 4 degrees Celsius. Everything else you can store at room temperature. The reagent refill pack contains the core reagents, including 10x CO2 indicator solution, debeading solution, and of course, algae beads. The full kit also includes the indicator color guides and all of the plastics required for the full lab. And don't forget the instructor's guide. There are a few important steps for preparing this lab. Start preparing reagents 3 days before you run the investigation. That allows the CO2 indicator to equilibrate to the surrounding atmosphere. This step is essential to running a successful investigation.
Don't skip this step.
To prepare your 1x CO2 indicator, mix 25 milliliters of 10x CO2 indicator with 225 milliliters distilled water. Make sure you use distilled water, since the chlorine in tap water will kill the algae. Bottled spring water will also work, as long as it's not chlorinated. Loosely cover and store the solution overnight or for up to 3 days to allow for air exchange. Be sure to use a container with a large surface area such as a beaker or cup. After equilibration, check the indicator again to make sure its pH is in the 7.9 to 8.3 range. 2 days before the lab, you will need to dispense and activate your algae beads. Without activation, the beads won't work.
Activate the beads.
Dump the algae beads in a clean cup so they're easier to see, and put the beads you don't use back in the bottle along with the storage solution. You can make a scoop by cutting a transfer pipette on the diagonal. Use the tip of another transfer pipette to transfer the beads to a cuvette. Use a transfer pipette to return any excess storage solution back to the beaker. Add 2 milliliters of distilled water. Incubate for 5 minutes to allow for the storage solution to diffuse out of the beads. Residual storage solution will interfere with both the debeading and CO2 indicator solutions.
Don't forget to wash your beads.
Discard the wash solution and add 2 milliliters of 1x CO2 indicator solution. The indicator solution that you use to activate the beads does not need to be air equilibrated. Cap the cuvette tightly and place it under a light source for 12 to 16 hours to get them photosynthesizing. There are a variety of lamps that you can use, but as a general guideline, when using a 60-watt fluorescent or LED bulb, place the cuvettes on their sides in a single layer. 15 to 25 centimeters away. Remember to use a light that is not too hot or bright. After activation, any algae beads that are white or yellow were probably exposed to too much heat or light and are not functional anymore. After activating the algae beads under light, rest them at room temperature in ambient light for 6 to 8 hours. This allows for photosynthesis to downregulate so that your students can see both photosynthesis and cellular respiration in a single lab session. You are now ready to start the investigation. The first steps of the investigation involve dispensing algae beads and replacing the CO2 indicator. If you are pressed for classroom time, you can do these steps yourself right before class. There are several different time frames for dispensing, activating, and resting the algae beads outlined in the Instructor's Guide.
Hey, what is this?
The Instructor's Guide I just mentioned. Bio-Rad's Photosynthesis and Cellular Respiration Kit is unique because it enables your students to study both processes in a single lab activity. Students use an inquiry-based approach to investigate the effects of light color, light intensity, and temperature. You can tailor the type of inquiry to your students' needs and interests. They can even create a mini ecosystem with aquatic snails. While this lab was designed for AP Biology, it fits with many life science courses including microbiology, ecology, environmental science, and agricultural science. And don't forget, the algae beads are reusable for multiple classes. Check out the Instructor's Guide for more information on this.
I don't get it. Why won't this turn on?
How did you get that again? For more information and resources about Bio-Rad's Photosynthesis and Cellular Respiration Kit, visit our website